Hi viewers, after a short gap, welcome back again to one more other video session from this channel. So in this video session, we will be studying regarding the structural organization of plastids, ribosomes, cilia and flagella and as well as nucleus in detail. So at first, if we consider plastids, so here what are plastids? Plastids are the subcellular organelles which are most commonly observed in the cytoplasm of certain living organisms like plants and as well as eukaryotes. Is it clear? So let me write it here. Plastids are present in plants and as well as in case of eukaryotes. And these specific plastids, they are absent in case of bacteria. So at first, if we consider the particular plastids, these particular plastids, they can be easily visualized under microscopic observations. As the particular plastids, as they are larger in size, these can be easily visualized under microscopic observations. And the particular plastids, they are enriched with certain pigments. So here, because of the presence of certain pigments, these particular pigments, they are responsible for imparting one specific color to the plastids. So here, basing on the presence of the pigments, so here we can classify the plastids into three different types, and these include chloroplast, chromoplast and as well as leucoplast. So let me write it here. Chloroplast, chromoplast, and the other one is leucoplast. So at first, if we consider the chloroplast, so here as the name itself suggests, chloroplast, it is the specific plastid which is enriched with a specific pigment which is nothing but what? Chlorophyll. Chlorophyll is the specific pigment which is responsible for trapping the sunlight and converting the sunlight into the chemical energy in order to synthesize the specific food molecules like sugars during the photosynthesis mechanism. And if we consider the specific chromoplast, so here as the name itself suggests, these are the particular plastids which are enriched with certain pigments like carotenoids and as well as xanthophylls. So here, because of the presence of these specific pigments, these particular pigments, they are responsible for imparting the yellow, orange and as well as red color. Is it clear? Because of the presence of certain pigments like carotenoids and as well as xanthophylls, these particular pigments, they are capable to impart certain colors like yellow, orange and as well as red. And if we consider the particular leucoplast, as the name itself suggests, leucoplast, these are the colorless plastids. They won't possess or they won't contain any specific color. And here the particular leucoplast, they are responsible only for the storage of food materials. So here, basing on the storage of the food materials, here we can classify the particular leucoplast into three different types, and these include amyloplast, ilioplast and the other one is erythroplast. So let me write it here. Amyloplast, ilioplast and the other one is erythroplast, or it is otherwise known as proteoplast. So at first, if we consider the amyloplast, as the name itself suggests, this is the specific leucoplast which is capable to storage the specific food molecules which are nothing but the sugars. So the reason why here the particular plastids, they are termed as what? Amyloplast. Is it clear? Because of the storage of the specific food molecules like sugars, here the particular leucoplast, they are termed as what? Amyloplast. Then in the same way, if we consider the specific ilioplast, so here the particular ilioplast, they are responsible for the storage of oils and as well as fats. So the reason why here they are termed as what? Ilioplast. And if we consider erythroplast, these are the specific plastids or these are the specific leucoplasts which are capable to store the specific protein molecules. Because of the storage of the specific protein molecules, these particular leucoplasts, they are termed as what? Erythroplast, or in other terms we can consider it as proteoplast. Is it clear? So let me write it here. Amyloplast, it stores food molecules like sugars. Then in the same way, if we consider the ileoplast, they store oils and as well as fats. And erythroblast, they store proteins. And here, in order to understand the structural organization of plastid, here we can take the advantage of the structural organization of chloroplast. So let me write it here. Chloroplast. So here, in order to understand the structural organization of plastid, here we can take the advantage of the structural organization of chloroplast. So here, the particular chloroplasts are the specific plastids which are most commonly observed in the mesophyll region of the plant body. Is it clear? Within the mesophyll region of the plant body, here we can observe the specific plastids which are nothing but what? Chloroplast. And here the size, shape and as well as the number of the chloroplast, it remains vary. Nearly in case of all the living organisms, here the size, shape and as well as the number of the chloroplast, it won't remain similar, it remains vary. So at first, if we consider the size of the chloroplast. So if we consider the dimensions of the particular chloroplast, the length of the chloroplast, it relates around 5 micrometers to 10 micrometers in diameter. And if we consider the width of the chloroplast, it relates around 2 to 4 micrometers. Width, it relates around 2 micrometers to 4 micrometers. Then in the same way, if we consider the shape of the chloroplast, it can be either discoidal shape, oval shape and as well as spherical shape. Is it clear? And even the number of the chloroplast, it also remains vary in case of the various living organisms. So here we need to understand one key point. Though the size, shape and as well as the number of the chloroplast, it remains vary in case of the various living organisms, but ultimately the function of the specific chloroplast, it remains similar nearly in case of all the living organisms. Is it clear? In case of all the living organisms where the specific chloroplasts are present, the particular function of the chloroplast is to store the food molecules, and even the particular chloroplasts, they are responsible for trapping the specific sunlight into the chemical energy in order to synthesize the specific food molecules, which are nothing but what? The sugar molecules. Is it clear? And if we consider the structural organization of the particular chloroplast, basically the particular chloroplast, it is a bilayered membrane. That means likewise the mitochondria, even the particular chloroplast, it has two specific membranes, and these include outer membrane and the other one is inner membrane. Is it clear? So here it is nothing but what? The outer membrane and the other one is inner membrane. And in addition to the outer membrane and as well as inner membrane, very adjacent to the inner membrane, here we can observe a specialized region which is present very adjacent to the inner membrane of the chloroplast, which is nothing but what? Stroma. Is it clear? Stroma of the chloroplast, it acts as a hotspot for organizing the dark reaction, and in addition, stroma of the chloroplast, it contains various enzymes which are responsible for synthesizing proteins and as well as carbohydrates, and in addition, stroma of the chloroplast, it contains double-stranded circular DNA, and even the stroma of the chloroplast, it contains 70S ribosomes. So here, because of the presence of the double-stranded circular DNA and as well as the 70S ribosomes, the particular chloroplasts, they are capable to synthesize their own proteins. That means without taking the advantage of nucleus or without the supervision of the nucleus, the particular chloroplast, they are capable to synthesize their own proteins, because the particular chloroplast, it contains its own double-stranded circular DNA, and even the particular chloroplast, it contains its own 70S ribosomes. So here, because of the presence of own double-stranded circular DNA and as well as 70S ribosomes within the stroma of the chloroplast, so here the specific chloroplast, it is capable to synthesize its own protein. So the reason why, likewise the mitochondria, even the specific chloroplast, here it is termed as what? Semi-autonomous organelle. Is it clear? So let me write it here. Chloroplast is termed as semi-autonomous organelle. Semi-autonomous organelle. And very adjacent to the particular stroma, here we can observe thin flattened tubular sac-like structures which are nothing but what? Thylakoids. Is it clear? Very adjacent to the stroma, here we can observe thin flattened tubular sac-like structures which are nothing but what? Thylakoid. On the surface of thylakoid, here we can observe many number of piles of coins-like structures which are stacked upon each other, and here they are termed as what? Granum or grana. Is it clear? On the surface of the thylakoid, here we can observe many number of piles of coins-like structures which are stacked upon each other, and here they are termed as what? Granum or grana. So here in what way the particular stroma, it acts as a hotspot for organizing the dark reaction, in the same way the particular granum or grana, it acts as a hotspot for organizing the light reactions. Is it clear? And here the specific granum or grana which is present on the surface of the thylakoid, it is capable to associate, or it is capable to interconnect with the neighboring or with the adjacent thylakoid grana or granum by taking the advantage of a specialized region which is nothing but what? Stroma lamellae. So here the specific granum or grana which is present on the surface of the thylakoid, it is capable to associate, or it is capable to interconnect with the neighboring granum or grana by taking the advantage of stroma lamellae. Is it clear? So here this is regarding chloroplast. Then after we will be studying regarding the structural organization of ribosomes. So let me arrange the things first. Then after we will be studying regarding ribosomes. So here what are ribosomes? So here ribosomes, these are the granular structures which was first identified or which was first observed by George Peled under microscopic observations. So here basically or chemically the specific ribosomes, they are ribonucleoproteins. That means they are chemically composed of RNA and as well as proteins. So the reason why here ribosomes, here they are termed as what? Ribonucleoproteins. Is it clear? So let me write it here. Ribosomes are ribonucleoproteins. Ribonucleoproteins. And here the specific ribosomes, they are present both in case of prokaryotes and as well as in case of eukaryotic cells. So here though the specific ribosomes, though they are present or they are observed both in case of prokaryotic cells and as well as in case of eukaryotic cell, but here the specific ribosomes which are present in case of prokaryotes, they are quite distinct from the specific ribosomes which are present in case of eukaryotes. So here in case of prokaryotes, the specific ribosomes which are present here, they are termed as what? 70S ribosomes. So let me write it here. In case of prokaryotes, the specific ribosomes which generally do exist, they are termed as what? 70S. But whereas in case of eukaryotes, the specific ribosomes which generally do exist, here they are termed as what? 80S. And here 70S ribosomes, basically they are composed of two specific subunits, and these include 50S and the other one is 30S. 50S and the other one is 30S. Then in the same way, the specific 80S ribosomes which are present or which are observed in case of eukaryotes, it contains two specific subunits, and these include 60S and the other one is 40S. So here S stands for what? Swedberg unit, which has obtained during the sedimentation coefficient of ultracentrifugation mechanism or the ultracentrifugation process. Is it clear? So here the function, the basic function of ribosomes is to organize the protein synthesis. The basic function of ribosomes is to organize protein synthesis. Right? Is it clear? So here then after we will be studying regarding cytoskeleton. So here what is cytoskeleton? So here let me arrange the things first. Cytoskeleton, right? If we consider cytoskeleton, so here what is cytoskeleton? So here the meaning of cyto is cell and the meaning of skeleton is the thing which provides one particular shape. Humans are having one particular shape because of the presence of what? Human skeleton. And in the same way, all the biological cells, they are having one particular shape because of the presence of what? Cytoskeleton. So here, cytoskeleton, it is an association of enormous number of proteinaceous filaments which are most commonly suspended within the cytoplasm of a biological cell. And here basically the particular cytoskeleton, it is composed of three differentiated components and these include actin filaments or microfilaments, intermediate filaments and as well as microtubules. So let me write it here. Cytoskeleton is an association of enormous number of proteinaceous filaments. And basically the cytoskeleton, they are composed of three differentiated components and these include actin filaments or microfilaments. Is it clear? And the next one is intermediate filaments. And the next specific component is microtubules. So here all these three are the specific components of the cytoskeleton. So here all these three specific components, they play an important role for providing the mechanical support to the cell and even they play an important role in case of the cell motility or the cell movement. And in addition, even the particular cytoskeleton, it plays an important role for the successful movement of chromosomes during the cell division. Is it clear? So here this is regarding cytoskeleton and then after we will be studying regarding cilia and as well as flagella. So let me arrange the things first. Then after we will be studying regarding cilia and as well as flagella. So here what is cilia and as well as flagella? Cilia and flagella, they are the locomotory organelles which play an important role in inducing the locomotion. So here for the successful locomotion of the particular biological cell, here the particular biological cell, they take the advantage of cilia and as well as flagella. So here cilia and flagella, they are the hair-like outgrowths which are most commonly present on the outer surface of the biological cell. And even the particular cilia and as well as flagella, they play an important role for the successful movement of the biological cell and even it plays an important role for the movement of the fluid-like component. Is it clear? So here the specific cilia and as well as flagella, they are present or they are observed both in case of prokaryotes and as well as in case of eukaryotes. But here we need to understand one key point. Though the specific cilia and as well as flagella, they are present both in case of prokaryotes and as well as in case of eukaryotes. In comparison with cilia, the particular flagella, it is comparatively longer. And in addition, here there is one more other difference in between the cilia and as well as flagella, which is observed in case of prokaryotes and as well as in case of eukaryotes. So here in comparison with the prokaryotic cilia and as well as flagella, in case of eukaryotic cilia and flagella, here we can observe a specialized arrangement which is nothing but what? Axoneme, which is composed of 9+2 arrangement. Here the particular cilia and flagella, though they are observed nearly in case of all the living organisms, in comparison with cilia, the particular flagella, it is comparatively longer. And in addition, the particular eukaryotic cilia and as well as flagella, it remains quite distinct from the specific prokaryotic cilia and as well as flagella. Here in case of eukaryotic cilia and as well as flagella, basically they are composed of a specific arrangement which is nothing but what? Axoneme. Axoneme, it is composed of 9+2 arrangement. So here what is axoneme here? Axoneme, it is basically composed of 9 doublet microtubules. Is it clear? On the peripheral region of the axoneme, here there is an availability of 9 doublet microtubules, and all the 9 doublet microtubules, they are enclosed or they are protected by the means of what? Plasma membrane. On the peripheral region of the axoneme, here there is an availability of 9 doublet microtubules, and here one particular doublet microtubule, it is capable to associate with the neighboring doublet microtubule by taking the advantage of what? Inter-doublet bridge, and even by taking the advantage of linker proteins. So here the linker proteins and as well as the inter-doublet bridge, it plays an important role in interconnecting the neighboring doublet microtubules. Is it clear? And in addition to this, on the interior side, here we can observe a pair of singlet microtubules. So here the pair of singlet microtubules, they are enclosed or they are protected by the means of what? Central sheath. Is it clear? So here we need to understand one key point. So here the 9 doublet microtubules, they are enclosed, or here the 9 doublet microtubules, they are capable to associate with the central sheath by taking the advantage of what? Radial spokes. So here the radial spokes, they play an important role in interconnecting the 9 doublet microtubules with the particular central sheath, which is responsible for enclosing or for protecting the pair of singlet microtubules. Is it clear? So here it is the specific arrangement which is most commonly observed only in case of the eukaryotic cilia and as well as flagella. Is it clear? So here the specific eukaryotic cilia and as well as flagella, or even in case of the prokaryotic cilia and as well as flagella, most commonly they originate likewise the centrosome from the basal surface of the body. In what way? The particular centrosome, it resembles its own native shape. In the same way, the particular cilia and as well as flagella, it resembles the shape of centrosome from the basal surface of the body. Is it clear? So here this is regarding cilia and as well as flagella. Then after we will be studying regarding centrosome. So here what is centrosome? So let me arrange the things first. Then after we will be studying regarding centrosomes. So here what are centrosomes? So here centrosomes, these are the specific subcellular organelles which are most commonly present within the cytoplasm of certain eukaryotic cells. Is it clear? So here centrosomes, basically they are composed of two cylindrical like structures which are present very perpendicular to each other, and here these specific cylindrical like structures, they are termed as what? Centrioles. So let me present it here. So here what is centriole? So here it is nothing but what? The centriole, right? Is it clear? Centrosome. Centrosome, they are composed of two cylindrical like structures which are present perpendicular to each other, and here each particular centriole, they are composed of 9 triplet microtubules. So here they are nothing but what? The 9 triplet microtubules, right? 9 triplet microtubules. And here this is nothing but the 9 triplet microtubule. So here all these are nothing but what? The 9 triplet microtubules. So here the 9 triplet microtubules which are present on the peripheral region, they are capable to associate with the proteinaceous hub by taking the advantage of what? A specific protein which is nothing but what? Radial spoke. So here all the 9 triplet microtubules, they are capable to associate with the proteinaceous hub which is present at the center by taking the advantage of a specific protein which is nothing but what? Radial spoke. So here the particular centrioles, they play an important role in forming the basal body of cilia and as well as flagella. As I have already mentioned you, the particular cilia and as well as flagella, they are capable to originate from the basal surface of the body by taking the advantage of what? By taking the advantage of centrioles. So let me write it here. Centrioles are composed of 9 triplet microtubules. 9 triplet microtubules. centrioles gives rise to cilia and as well as flagella. Is it clear? The particular centriole, it plays a prominent role in forming the basal body of the cilia and as well as flagella, and even the particular centriole, it plays an important role in regulating the cell division. Centrioles are responsible in regulating cell division. And here centrosome, it is a specific subcellular organelle which is present only in case of animal cell. So here though the specific plant and as well as animal, they come under the eukaryotic cell organization, but here the specific animal, the particular animal cell, it remains quite distinct or it remains vary from the particular plant cell in one of the particular aspect, which is nothing but what? Presence of centrosome. The particular centrosome, it is present only in case of the particular animal cells, and here they are absent in case of plant cells. So here the particular centrosome, it plays an important role in regulating the cell division. Is it clear? So here this is regarding centrosome. Then after we will be studying regarding the structure of nucleus. So let me arrange the things first. Then after we will be studying regarding the structural organization of nucleus. So here what is nucleus? Nucleus, it is a round spherical ball-like structure which is responsible for controlling and coordinating the various life activities of a particular biological cell. That means in what way for controlling and coordinating the various life activities within the particular living organism. Here the controlling system or the coordinating system, it plays an important role. In the same way, for controlling and coordinating the various life activities within the particular biological cell, here the nucleus, it plays an important role. And basically, during the interphase stage of the cell cycle, the specific nucleus, it has four differentiated components, and these include nuclear membrane, nucleoplasm, chromatin material, and as well as nucleolus. Is it clear? So here all these are the four differentiated components of the particular nucleus during the interphase stage of the cell cycle. So here the outer membrane of the particular nucleus, as I have already mentioned you, the interphase stage of the cell cycle, it has four differentiated components of the nucleus, and these include nuclear membrane, nucleoplasm, chromatin material, and as well as nucleolus. So here basically the specific nuclear membrane, it has two differentiated layers, and these include outer nuclear membrane and the other one is inner nuclear membrane. So here the outer membrane or the outer nuclear membrane, it remains in association with the surface of endoplasmic reticulum, and chemically the outer membrane of the particular nucleus, it remains very similar with the chemical composition of the endoplasmic reticulum. On the surface of the nucleus, in general, endoplasmic reticulum is present. So here the chemical composition of the peripheral region of the endoplasmic reticulum, it remains very similar with the outer nuclear membrane. Is it clear? And in between the outer nuclear membrane and as well as inner nuclear membrane, here there is an availability of specific space which is nothing but what? Perinuclear space. Is it clear? And in between the two successive nuclear membranes, here we can observe specific minute pore-like structures which are nothing but what? Nuclear pores. So here what is the function of nuclear pores? So here the particular nuclear pores, it plays an important role for the successful organization of exporting and as well as importing of the various molecules. That means, as we all know, nucleus, it acts as a hotspot for the successful synthesis of RNA. So here after the successful synthesis of RNA within the particular nucleus, the specific RNA, it has to export from the nucleus to the cytoplasm. In the same way, within the particular cytoplasm, protein synthesis takes place. So here after the successful synthesis of protein within the cytoplasm, now the specific protein, it has to import from the cytoplasm into the nucleus. So here for the successful organization of exporting and as well as importing of the various molecules, here the particular nucleus, it takes the advantage of minute pore-like structures which are nothing but what? Nuclear pores. Is it clear? And then after we will be studying regarding nucleoplasm. Nucleoplasm. And initially the specific nucleus, for the very first time, it was identified or it was observed by Robert Brown under microscopic observations. And here if we consider the components of nucleoplasm, Basically, it is composed of two differentiated components. These include chromatin material, and the other one is nucleolus. So at first, chromatin material. So here, what is chromatin material? So here, the specific chromatin material, it can be successfully differentiated into two different types, and these include heterochromatin and the other one is euchromatin. During the interphase stage of the particular cell cycle, here the specific chromatin material, it remains so loose form. So the reason why here it is termed as what? Euchromatin. So here euchromatin, it is the specific chromatin material which is most commonly observed during the interphase stage of the particular cell cycle. In the same way, during the mitotic stage of the particular cell cycle, here we can observe tightly formed or tightly condensed form of the specific chromatin material, which is nothing but what? Heterochromatin. Heterochromatin, it is observed during the mitotic stage of the particular cell cycle, but whereas euchromatin, it is observed during the interphase stage of the particular cell cycle. Is it clear? Heterochromatin is observed during mitotic cell division. Euchromatin is observed during interphase stage of cell cycle. So here within the specific cell division, here the specific chromatin material, it won't remain in loose form. So here within the particular interphase stage of the cell cycle, the specific chromatin material, it remains in loose form, but during the specific mitotic cell division, the specific chromatin material, it won't remain in loosened form. Here the specific chromatin material, it remains successfully packed within the particular chromosomes. Chromosomes are the condensed form of chromatin material. So here the specific chromatin material, it won't package within the particular chromosome directly. At first, the specific chromatin material, it is successfully wrapped around the histone octamer. So if you look at here, easily we can understand. So here it is nothing but what? The histone octamer, right? On the surface of the histone octamer, in general, the specific chromatin material or the specific DNA content, it is successfully wrapped here. So once when the specific DNA content, when it is successfully wrapped around the histone octamer, now here it leads to the formation of what? Nucleosome. So here it is nothing but what? Nucleosome. So once when the specific DNA, when it successfully wraps around the histone octamer, now here it leads to the formation of nucleosome. Is it clear? So here, basing on the position of the particular centromere, here we can classify the specific chromosomes into 4 different types. Is it clear? If we look at the specific chromosome, so here it is nothing but what? The structure of the linear chromosome, right? So here the two arms of the chromosome, they are capable to associate together by taking the advantage of a specialized center region, which is nothing but what? Centromere. So here, basing on the position of the centromere, here we can classify the chromosomes into 4 different types. So let me write it here. Basing on the position of the centromere, here we can classify the specific chromosomes into 4 different types, and these include metacentric, submetacentric, and the other one is acrocentric, and the last one is telocentric. So at first, metacentric. So what is metacentric? So once when the specific centromere, when it remains at the center. Under such condition, the two arms of the chromosome on either side, they are equally differentiated, and here it is termed as what? Metacentric. And after, what about submetacentric? So here what exactly happens in case of the submetacentric? In case of submetacentric, the specific centromere region, instead of remaining at the center, it moves slightly far away from the center. Is it clear? In case of metacentric, the specific centromere region, it remains at the center. So the reason why the two chromosomal arms, they are equally differentiated. But whereas in case of the submetacentric, the specific centromere region, instead of remaining at the center, it moves slightly far away from the center. So the reason why here it is termed as what? Submetacentric. That means the two chromosomal arms, they get successfully differentiated, and the other two chromosomal arms which are present at the upper region, they are not successfully differentiated, and here it is termed as what? Submetacentric. Is it clear? Then what about acrocentric? So here in comparison with submetacentric, in case of acrocentric, the specific centromere region, it moves some more far away from the center, and here it is termed as what? Acrocentric. Then what about telocentric? So here the meaning of telo is the end part. So here during the telocentric, the specific centromere region, it moves completely far away from the center, so that the two chromosomal arms, they are equally differentiated, and another two chromosomal arms, they are not at all differentiated, right? They remain at the end part, and here it is termed as what? Telocentric. So here this is regarding, basing on the position of the centromere, here we can classify the particular chromosomes into four different types, and these include metacentric, submetacentric, and as well as acrocentric and telocentric. And here on the peripheral region of the particular chromosome, here we can observe a specialized region which is not at all visualized under staining procedures, and here it is termed as what? Secondary constriction or satellite. Occasionally, in case of few chromosomes, here we can observe a specialized secondary constriction or satellite which is not visualized under staining procedures, and here it is termed as what? Secondary constriction, or in other terms we can consider it as satellite. Is it clear? And after we will be studying regarding microbodies. So here what are microbodies? So here prior to the explanation of microbodies in detail, here I need to mention you one key point. So here during the explanation of chromosomes, here I forgot to mention you one key point. On the surface of the chromosomes, here there is an availability of a specific disc-shaped protein complex which is nothing but what? Kinetochore. So here by taking the advantage of the specific kinetochore, which is a disc-shaped protein complex, the specific chromosomes, they are capable to align with the particular spindle fibers during the cell division. Is it clear? And if we consider the particular microbodies, so here these specific microbodies, they are minute vesicle-like structures which are present within the cytoplasm of plant cell and as well as animal cell. Is it clear? So let me write it here. These are minute vesicle-like structures. And here they are present in plants and as well as in case of animals. So here the specific microbody which is present in case of plants, here they are termed as what? Glyoxysome. And here the specific microbody which is present in case of animal cells, here they are termed as what? Peroxisomes. So here both the particular glyoxysome and as well as peroxisome, they has their own enzymes. Is it clear? So here this is regarding plastids, ribosomes and as well as nucleus in detail. And in the next upcoming video session, we will be studying regarding biomolecules. Is it clear? So here I hope that this video will help you a lot. So if you like this video, just hit the like button and share it to your friends. Thank you.